All right, so I'm coming from the Max Planck Institute for Biological Intelligence. Was there a question? Sorry? Was there a question?
Oh, no, do you have a microphone? Or...
I do.
Oh, right. Sorry. Yeah.
I do.
Yeah.
Can you hear me?
Yeah.
I thought it was okay. All right. So Max Planck Institute for Biological Intelligence, we've reinvented ourselves. We used to be the Max Planck Institute of Neurobiology, and we refounded the institute as the one for biological intelligence. We're interested in how biological organisms solve problems, clearly looking at biological intelligence as a counterpoint to artificial intelligence. And we're interested both in short timescales, cognition, thinking, navigation, finding food, finding mates, what the brain can contribute to that, but also in longer time scales, like evolutionary processes. So my lab is particularly interested in fish behavior, and I was very happy to hear Bruno talk about the jumping spiders, which have miniature brains, and they solve really complex problems, and they do so in a super efficient way. Vertebrates also are not so bad. So here we are watching from below. This I lifted from Wired magazine. So we're filming here from below a minnow that is frolicking in the pond, and unbeknownst to this fish, there's a predator, a kingfisher, snatching this fish out of the lake. And this is what we are witnessing here is kind of an anecdote or an episode in an evolutionary arms race between two visual systems, that of the kingfisher that is picking prey from high above, and that of the fish that is, in this case, losing this battle, but whose visual system has evolved to be very good at escaping from an approaching predator. So there has to be kind of a continuous line of ancestors of this fish that did pretty well and did escape predation before they could reproduce. So our lab is interested in kind of a very small fish, a zebrafish larva. And here now in this video, the tables are turned. Now the predator is the teleost fish, and the unwitting victim is a paramecium, a single-celled organism. The fish is fixating on and is approaching and likes to come from below, is approaching and is then catching. So, and this is just sort of the last snapshot of the life of a paramecium as it disappears in the mouth of the predator. So we can, in the lab, recapitulate these behaviors in a head-fixed animal. So this is work that goes back now 2 decades. So here my postdoc Julie Semmelhead, when she was in the lab, had fun moving a little white dot in front of a head immobilized fish. The fish goes crazy and likes to catch it. And in fact, these kinematic motifs that we can isolate here very closely resemble those that you see in a free-swimming fish. We know this fish is trying to catch prey. And when we change the stimulus and we display it on the side, and we have now a dark dot that is looming, the fish also shows a behavior which is indicative of an attempted escape movement. So these preparations have now been used by our lab and now also by several others, many others in fact, as a way to image the activity in the brain of these animals. They have transparent brains, and we can, using also genetic tools, we can identify the cell types and the circuits that are involved in recognizing and perceiving these different visual stimuli. So today I'm going to do something that I would never advise a student to do. I'm going to tell you a lot of small stories rather than focusing on one big single story, because I think in this room there are many people who are maybe not so familiar with this system, so I'm going to give you lots of flavors of what we can do in this system. And I also want to advertise it, because I think we have now collected a lot of ground truth about the zebrafish brain and about the visual system in particular. So there aren't that many free parameters, not so many degrees of freedom. So modelers can go now crazy on this system and do biologically plausible modeling and theorizing. So today I will sort of focus a little bit on this one problem that I find very interesting. And that is that the same identical stimulus can lead to different behavioral outcomes. Okay. So this is what I call flexibility as opposed to plasticity. So I'm not going to talk about plasticity today. I'm going to talk about flexibility, the repurposing of a circuit for different behaviors in a context and otherwise dependent manner. And I will focus on the optic tectum of the fish. So this is this visual motor hub in the middle of the brain of the fish, receives retinal inputs, processes this information and transforms it into motor output. And we know a lot about the tectum. Last year, I talked about the cell types of the tectum and biological principles of how cell types are made and wired up. And today I will talk about also connectivity, but maybe with more of focus on behavior. Okay, so zebrafish, I think, offer a unique opportunity to dissect the computational principles and also the neuronal and neurobiological implementation of these principles. So first I will show you how robust this visual system is. It is to a large extent hardwired. And we start with the retinal ganglion cells. There are the usual number of several dozen types, so indicated here by the colors. And they come in these different morphological shapes. So these are the dendritic arborizations. We've done a survey of really now thousands of these ganglion cells. You can put them into different classes. And there are some really funky ones, like this one here, which looks like a upside down girl in a dress with a very large foot. And then you also have your bistratified, monostratified, and diffuse types. And we know from the work of many labs, including ours, but certainly not exclusively ours, also from work in mouse and chicken and monkeys, that these retinal ganglion cell types represent feature detectors. So they are interested in particular features in the visual scene, such as movement, direction of movement, contrast, color, etc. And these different types then combine all these features in the optic nerve, but keep the features separate because each axon carries on information about only one feature. And they then project into the brain. So here I've labeled in the optic tract of the retinal ganglion cells, formed by the retinal ganglion cells, as they terminate in 10 arborization fields, which are little neuropil areas that have Latin names and correspond to the well-known central visual areas. And so there's all the way from hypothalamus to prethalamus, thalamus, and pretectum. There are different arborization fields, and then almost all retinal ganglion cells, with a few exceptions, terminate in the tectum. And here they terminate one axon in one layer, 1 out of 10 layers in the tectum. So this is an old rainbow labeling experiment where we visualize the projections of about 20 retinal ganglion cell axons, and when you sort of image from the top, it all looks like a tangled mesh, but then you turn the confocal image stack on the side and you see these beautiful layers formed by the retinal ganglion cells. So we've learned a lot about the layers in the tectum, so they are shown on the right side. the arborization fields and pretectum, etc., are shown on the left, and the morphologies of the rectangular ganglion cells are shown on the left side. Let me see if that cursor. Oh, here it comes. Here comes the cursor. Okay, was hiding so far. So here are the shapes of the rectangular ganglion cell, and you can see that each of these lines, so there's 20 of these visual pathways, anatomically defined, is composed of a combination of rectangular ganglion cell shapes. So each of these lines combines different features represented by the rectangular ganglion cell types, and they project into different layers of the tectum, and often they make collaterals here in these arborization fields. So Yvonne Kölsch, when she was in the lab, did a transcriptomic survey of these rectangular ganglion cell types and was able to also make transgenic lines using CRISPR-Cas genome engineering, and we could show for different molecular types of rectangular ganglion cells, they had very specific laminar specificities and particle specificities, and we were interested in finding the function of these different visual pathways, and for many we've succeeded. So for instance, this type here, the MAFAA ganglion cells, they respond specifically to prey, to the paramecia that I showed you in one of the first videos, and they project into an area that is called AF7 in the pretectum, and also in the superficial layer of the tectum. And when we ablate any of these components, either the ganglion cell type, or the AF7 neuropil, or the superficial layer in the tectum, the fish is unable to detect a prey. When we optogenetically stimulate this pathway, the fish shows movement of the tails as if there was a prey item in its environment. So it tries to capture an imaginary prey. I have to go faster. Okay. Other layers are specialized for other types of behaviors, and so we know quite a bit about that. With Karthik Shekhar, we have a CRCNS project where we look at the development of this complement of ganglion cells over developmental time. As the fish grows, there are new behavioral demands of the animal. So the positioning, a typical hunting episode works like this. The fish detects a prey item at a distance in its peripheral visual field, then turns towards it, swims towards it, and then captures it. So typically there's a small thing somewhere in the periphery, and then as the fish successfully approaches it, it becomes bigger, and it is now represented in the anterior tectum, in fact, on both sides of the brain. So when we look at the tectum and the representation of the prey goes from small and distant in the posterior tectum to close and large in the anterior tectum. And in fact, when we now image responses in the tectum, we find that, so this is an array of visual stimuli that we used, including prey stimulating objects. We find that there are cells, tactile cells, that are specialized to detect small things, and other tactile cells that are responding to moving. Those are completely different populations, right? So the processing of the stimuli is segregated anatomically, and then also there's a large fraction of cells that are responding to large objects. Now, when we look at the position of these cells, it is striking that the cells that respond to small objects are enriched in the posterior tectum. Remember, that's where the prey initially appears in the peripheral visual field, whereas those that respond to larger objects are in the anterior tectum. So the regional topic distribution of these prey-selective cells sort of follows the sensory ecology, the expectation from sensory ecology of the fish. So I will skip over this. This is our sort of connectomic now analysis of these circuitries. And it's largely descriptive, but it's beautifully consistent with our expectations. So we've now reconstructed with electron microscopy the ganglion cells that project to AF7 and to the tectum. So those are the prey detecting cells. We can reconstruct the postsynaptic connectome of single retinal ganglion cells in the tectum. And we also can show that there are premotor neurons that are perfectly consistent with their involvement in prey, orientation towards prey, bending the tail, opening the jaws, converging the eyes, etc. So let's do this. So what I would like to show you first now is the extreme robustness of this system to perturbations. So we did the most extreme that we could think of. We took a mutant that was lacking all retinal ganglion cells. So these are fish that have never seen through their retina a single, so that have never conveyed visual information to the brain. So the ganglion cells are not even born. So this is not just cutting the optic nerve. These brains have never had any visual information. Also, there's no factor secreted by the retinal ganglion cells that could somehow organize or assemble the neural circuits. There's also no retinal waves in these animals. And we then tested, first of all, how the cells differentiate in the brain, in the visual brain, and they do so apparently normally. We could not find any difference in the number of cell types, even in the relative proportions of these cell types. We then went in with the optogenetics and activated cells that elicited prey capture behavior in these entirely blind fish. We could activate prey capture circuitry. So our conclusion from this is that neither sensory experience, nor retinal waves, nor any ganglion cell-derived molecular factor has a detectable effect on the development of the visual brain in the zebrafish. So super robust. Still, this robustly wired system, okay, is flexible. Did you factor in that I had these technical issues?
Yeah,
Can I have maybe 2 minutes for the?
yeah, but you won't get any questions.
Okay, I'll check that because I really wanted to drive home that message that the system is also super flexible. So I'm now going to oscillate a little bit between the trivial and the profound. Number one, the same stimulus can be interpreted differently depending on its context. So as the fish moves itself towards the prey goes from small to big, and the fish will continue its approach. When the fish is sitting and the object is approaching, it may undergo the exact same stimulus trajectory. So the retinal representation of the stimulus will be identical. Still, the fish will escape. Second instance of a flexible system is that we showed a while ago that serotonin can actually switch the tectum from a prey detecting and a system that's interested in prey and approach sensitive to one that is rather escaping. And this is done by recruiting cells that are dormant using this neuromodulator system. And so serotonin is a hunger signal that modulates the visual processing by selectively increasing the number of prey detecting neurons. The third example of flexibility is one that one could call bottom-up attention. So when the fish is responding, is exposed to a looming stimulus, it will escape in the opposite direction, away from the stimulus. It does so also to weak stimuli. When you present two looming stimuli here in the top, it will respond to the more salient stimulus by escaping. When you show two equally strong stimuli, it will stochastically choose one direction. And we can show that this is due to a winner-take-all circuitry that connects the two tactile hemispheres. With that, I'd like to thank you for your attention and